Hello students, welcome to the part 2 of absorption by root. So let's continue our process. In the previous lecture we have completed imbibition. Okay, and I have given the examples also. Now we are going to start with a new concept, that is the diffusion. Here the word diffusion means scatter. Okay, in more easy sense we can write the word diffusion means spread also. Spread. Okay, spread means moving from one place to another. That's a easy word, right? Now I'm going to read the definition for you, and then we can start with the process. Diffusion is the free movement of molecules of a substance from the region of higher concentration to the region of lower concentration when two are in direct contact. Okay, so there is one example out here which is situated. This diffusion starts at your home. Okay, if you would notice, when your mom cooks up the food inside the kitchen and you are sitting in the hall, so after few minutes or hour, you would notice the smell which was there inside the kitchen suddenly comes in the hall as well. So this is known as the diffusion. Here the word substance, you have to be very specific. The word substance here means solid, liquid and gases. Okay, why the substance word is that important? Because when we are going to learn osmosis, at that period of a time you would come to know about the difference. Okay, right now just keep in your head, diffusion occurs in the plants in the form of a substance like solid, liquid and gases, which helps to transport up the water, minerals and the gases like carbon dioxide and oxygen. Okay, let's move to the next slide. Now this is an experiment of diffusion. As in the previous slide you have seen that diffusion can occur at home as well. So this is an experiment time. What I'm going to do, I'm going to insert a drop of a dye solution. As you can see, the hand which has a dye solution, we are going to insert a drop inside the beaker, and beaker is filled with the water. So we would notice when we insert up this dye solution inside this beaker which is full of water, the molecules of the dye solution get concentrated in one region. Okay, the molecules of the dye concentrate at one region. Okay, after few minutes, in the slide 2 you can see this part. The molecules in the first slide are more concentrated, and in the second slide if you would notice, the molecules of the dye solution got scattered or spread, you can say. Okay, and in the third slide, after few minutes or hours, you would notice that the dye molecules get evenly distributed. What do you mean by evenly distributed? Means it got involved with the water. So this shows the properties of a diffusion. If you would have noticed of the definition, the moment of higher concentration, as we can see in this condition, to the moment of the lower concentration. In this part and this part, if you can see, there is no dye molecules. Okay, but this was highly concentrated, so it shifted the region towards this part, and this leads to the third part. Okay, understood? Now osmosis. What do you mean by word osmosis? The word osmosis means the movements of the water molecule from a region of higher concentration to the region of lower concentration through a semi-permeable membrane. Okay, through a semi-permeable membrane. Here these words are very important. What is the word? That is the movement of the water molecules. Okay, and through a semi-permeable membrane. Right now I'm going to use semi-permeable membrane in a short form, that is SPM, but during exam time you have to write in a full form only. See this experiment, this sky blue color, okay, which is on my left hand side, is blue in color. That blue color indicates that these are the water molecules. And on my right hand side, if you could notice here in this part, there are less number of water molecules. Okay, osmosis can only occur in the water molecules, okay, from a region of higher concentration to the region of lower concentration. See, the water molecules on the left hand side is more, and the water molecules in the right hand side is very less, okay, very less and very more. So what is going to happen once we start up this experiment, this water molecules, if you can see with this arrow, are going to move into this region, okay, are going to move into this region. And what do you mean by this word semi-permeable membrane? You have heard this word before also in cell chapter, that the cell membrane is semi-permeable in nature. The word semi-permeable, semi means half and permeable means allowed, okay, word permeable means allow. It is going to allow only certain substances, like water can move from this solution to this solution. But if you are going to see this big circles, let's assume that this is a sugar solution, okay, these are the sugar molecules. This won't be moving in that side. Why? This is not going to occur. Why? Because the molecules of the sugar are quite big in nature, and there is one line in between. There is one line in between with a small gaps. There is one line in between with a small gap like this. So it is only going to allow the substance which are small in nature, okay, it is not going to allow the substance which are big in nature. So that is why the water molecules are small in nature. So it is going to shift on the right hand side, okay. So we got the meaning of the osmosis. The word semi-permeable membrane and water molecule is very important. Let's move to the next slide. Now this is an experiment time. Let's understand the importance of the semi-permeable membrane, okay. SPM, I'm going to use a shortcut, that is SPM only, semi-permeable SPM. See, there are the apparatus. Before starting of the experiment, I would like to start with some of the name of the apparatus. See, this one is a stand, okay, this is a beaker, we already know. Inside the beaker it is filled with the water, and this line, this apparatus name is thistle funnel, okay. So we got this part. This is a stand, this is a beaker, inside the beaker there is a water, and this stand is called thistle funnel. Now I'm going to read up the setup part for you. There is a sugar solution in the thistle funnel. What is the thistle funnel? I made you aware. This is the thistle funnel with cellophane paper on the mouth, where on the mouth means here, okay. In the second part, there is the sugar solution in the thistle funnel with a rubber sheet on the mouth here. Third setup, there is a sugar solution in the thistle funnel with muslin on mouth here, and water in the beaker. If in these three slides you would notice, the only difference was cellophane paper, rubber sheet and muslin, okay. There were only this difference. The whole statement is same. There is a sugar solution in the thistle funnel with dash on mouth, water in the beaker. Only the difference was cellophane paper, rubber sheet and muslin. Let's perform this experiment. Okay, suppose in the initial level the water was here. Okay, after an hour we would notice that the water inside the cellophane paper rise. I'm going to show it by arrow. The upward arrow means rise. Okay, the water, the solution inside the thistle funnel got raised. Why it got raised? Let's keep it on hold. There was a third experiment. Okay, second one, sorry. Rubber sheet. The initial level, initial means the starting level. After an hour the level remains the same. Okay, the level remains the same here only. In the third part, what happened? The initial level of the solution was here in the thistle funnel. After an hour the solution level got decreased. Okay, the solution level got decreased. See, I am going to repeat this part for you again. The solution in setup A got rise, the solution in setup B remains the same, and the solution in the beaker C got fall. Okay, not in the beaker, inside the thistle funnel it got decrease. So what would be the reason behind that? The difference I already mentioned in the start only. Cellophane paper, rubber sheet and muslin. These were the only three differences. This cellophane paper is semi-permeable in nature, which I am going to write by SPM. Okay, what do you mean by SPM? Semi-permeable membrane. As it was semi-permeable membrane, okay, I have already said that it is what is going to happen. The water molecule which was there in the beaker enter into this thistle funnel. Okay, got entered into this thistle funnel. So that is why the solution which was there inside the thistle funnel also got raised up. This was the first reason. Second, as I already said, in the second part, that is a setup B, there was no rise. Okay, initial level was this, and after an hour also it remains the same. Why there was a no change? Because on the mouth here it was protected with a rubber sheet, and the function of the rubber sheet was that it was impermeable in nature. Impermeable in nature. I am and permeable. Okay, impermeable in nature. What do you mean by word impermeable in nature? Impermeable means it is not going to allow any of the substance to enter or exit. Okay, the water which was there inside the beaker is not going to go up also. The solution inside the thistle funnel is not going to go down also. Okay, so that is why the level remains the same. Now this is the third part, setup C. With muslin cloth we have covered its mouth, right? So what happened in the setup C? The starting level was up. After an hour, what we would have noticed that the level of this part got fall. Okay, the solution which was there inside the thistle funnel got fall. Why it got fall? There is a reason behind that. Because see, this is a cloth, right? If you would notice, the pores in the muslins are quite big. Okay, the pores in the cloths are quite big in nature. So what is going to happen? The solution, okay, the solution inside the thistle funnel moves into the beaker. Okay, moves into the beaker like this. Okay, so that is why the solution which was there inside the thistle funnel got decreased, because the sugar molecules which were present inside this part got into this beaker, and because of this the solution, there was a fall in the thistle funnel. Okay, there was a fall in the thistle funnel. As the pores were big, the sugar molecules move out of this thistle funnel into the beaker. So that is why there was a fall in the level of the Thistell funnel. Okay. Endoosmosis and exoosmosis. So two type of osmosis that occurs in the cell. One is the endo and other one is the exo. We have already done this endo and exo concept. Okay, the word endo and exo. We have covered this endo and exo in the chemistry also. Okay, endo means in and exo means out, exit. Exit. What is going to happen? Water moves into the cell. This helps to swell the shell. Let's understand. Suppose I am going to take shriveled shell. What do you mean by shriveled? Which are not perfectly big in nature, which are small in nature. Okay. So I am going to take a shell which is shriveled. Okay, and I am going to put them into the solution. I'm going to put that into a solution. This is a beaker. Inside the beaker there is a solution. Okay, students, note that solution does not mean water. Solution means water with addition of something. I'm going to put the shells. After few hours I'm going to see the size. So this was the before part and this was the after part. So we could notice when the shells were shriveled up, it put into the solution, it got swelled up. Okay, this means endoosmosis occurred inside this cell. Exoosmosis, I'm going to come. If suppose I take this cell, okay, I am going to take this shell and I am going to put them in beaker which is filled with a solution. I already made the word solution very clear. I am going to put them in the solution. After few hours, I would notice the cells which we have put inside the beaker got shriveled up. Okay, so what would be the reason behind that? Think over it. Okay, so many of the students could have noticed that the difference lie within this beaker. Okay, why? Because this beaker was filled with the solution, even this beaker was filled with the solution, but they were filled with the different amount of solution. Now let's understand what are the different types of solution. So this means tonicity. So I'm going to read the definition for you, then I'm going to make you explain. Tonicity: relative concentration of solution that determine the direction and the extent of diffusion is called tonicity. Okay, relative concentration of the solution that determine the direction and extent of diffusion is called tonicity. Let's start. See, there are three types of solution: isotonic, hypotonic and hypertonic. Let's start with isotonic. You have heard this word in chemistry: iso, isotopes, isobars. The word iso means same. I'm going to take some cells and I'm going to put that cells in the beaker. Okay, I would notice after an hour the cell size remains the same only. Okay, how we came to know that the cell size remains the same? Look at this part. The size of the arrows is same. Okay, the size of the arrow is same. This indicates same. Okay, the solution was simple and very clear. The cell size haven't got increased nor it got decreased. It remains the same. So this is very easy because I have already made you learn the word iso and I have linked up with the chemistry topic, this word. Now we are going to take the next part, that is hypotonic. Hypotonic. Again in biology, you have heard this word hypo. For hypo means less. Word hypo means less. Where you have heard this hypo word? Hypo, hypotension, hypertension, low blood pressure, high blood pressure. Okay, in blood pressure terms you have heard this term, hypo. The word hypo means there is less of solute, and more of water. Okay, let's understand this part. Suppose I am talking about this part. Inside the beaker there was a hypotonic solution. Okay, the nature of the solution was hypotonic. The word hypotonic means less solute and more amount of water was present inside this beaker. Okay, so when I put cells in them, after few hours the cell inside the solution got swelled up. Swelled up means it got big in nature. So one can see that easily. See, this downward arrow indicates that there was more amount of water which got absorbed by the cells and only less amount of water get released out. So that is why the cell size got increased up. Okay. Now I am going to take the next example, that is the hypertonic. The word hypertension, high blood pressure. So what do you mean high blood pressure? More. What more? More of solute. This solution is more of solute and less of water. More of solute and less of water. So what we did, we took a beaker 3 and I filled with a hypertonic solution which was containing more solute and less amount of water. Okay, when we put the cells inside this solution, the cell size got shriveled up. So why did this happen? Because the cell was swelled up. I put it in the beaker which was hypertonic, and after few hours the water which was present inside this shell, the water which was present in the cell got exit out, and so that is why the cell size got shriveled up. As we can see in this part, this arrow, there was less amount of water intake and more amount of the water got exit. Okay, more amount of the water got exit and less amount of the water got absorbed. And here in this case, more amount of water got intake and less amount of the water got exit out.